Assalamu alaikum, dear student. In this lecture, that is related to your practical number 2, we will cover cytogenetics and karyotyping techniques used in genetics and particularly in animal genetics. Cytogenetics basically is the study of the chromosomes. So, cyto, we know it's related to the cell, and genetics, of course, the genetic material of the cell that is visible in a cell is the chromosome. And the cytogenetics deal not only with the number of the chromosome in each cell, but also with the structure, function of the chromosome, its behavior, particularly in relation to the inheritance of the genetic material or the genes, then its organization, and finally the expression that is mainly in the form of the chromosomes and in the form of the genes. Chromosome is basically a combination of two words, chromo and some. Chromo, that means the color body. First time it was seen as a color body after dyeing with certain dyes, so that's why they are named as chromo. And as they were color body, so they added the some, that means the body, so that became the name chromosome. We know the difference between eukaryotes and the prokaryotes. So in eukaryotic cell, the study of the chromosomes or the cytogenetic analysis, they are very important subject traditionally, and mostly these have been examined under the light microscope. Well, if we see the structure of the chromosome, you can see in this slide there are two arms on one side and two arms on the other side, while they are connected through a center that we call the centromere, uh, and it have kinetochores. This is the structure of the chromosome. Uh, Now, each of these structures or arms which are separated, these are called the sister chromatids. Well, how we can differentiate between gene, DNA, or the chromosome? So it's very simple. Basically, chromosome is a bigger body and it contains the DNA, and that DNA is the combination of different genes. This diagram elaborates more clearly. You can see a chromosome, and this chromosome is further, you can say, if we take a segment of this chromosome, then it have further parts which contains the molecular marker, the actual coding gene. Then there could be more than one molecular markers, and we take this a small fragment of the this part, the part second shown in this diagram. It would be further uh, down sequenced in the form of ATCG, which are the four nucleotides of the DNA. So basically, this DNA and a lot of these nucleotide bases, they join together, they form genes, they form markers, and when they are condensed, they get the shape of the chromosome. So while we analyze the chromosomes and the genes, there could be some genetic errors, and that could be because of the deletion or the insertion of the genetic material, that is the nucleotide bases, and there could be abnormal number of the whole chromosomes or the genes, or even could be misplacement of the single base. A single nucleotide base could be misplaced, and sometimes it could have a devastating effects just like we have seen in case of the hemophilia. And these genetic abnormalities can range, uh, which are harmless if they are not in the coding region on the codings, particularly on in the position where they cannot alter the amino acid profile of the protein. They could be harmless, or they could be very severe, like vitamin deficiencies, food allergies, cancer. We have seen in the theory class in the that one nucleotide base change in the gene of the uh, related to the hemophilia, it can cause the hemophilia. Similarly, cancer, the change in the P5 gene, it can result into the cancer. And this can be sometimes so serious, it can prove fatal for the individual. Or there could be some uh, by birth genetic defects. Now, what tissues are appropriate or suitable for the chromosomal studies? So, ideally, in a normal cell which is not in the dividing phase, we cannot see the chromosomes. So, we need any cell which is dividing to study the chromosome, because during the division of the cell, the chromosomes they are condensed and they could be seen under the microscope, as we have seen in the previous practical. So this can be done in vitro in the petri dish. Cells could be incubated or they could be stimulated to grow, to multiply, and during that process of multiplication, the chromosomes could be condensed and we can see under the microscope. So, under the light microscope, distinct chromosomes could be seen during the cell cycle, which we have already discussed during the mitosis of the cell or division of the cell. So, after culturing in vitro, proportion of cells are arrested in mitosis, in mitotic phase or in mitosis, and then they are harvested, they are separated for chromosome analysis. After harvesting the cells, preparations are dropped onto the glass slides and stained. We have seen in case of the uh, grasshopper example studied in the last lecture of the practical. So mostly in mammalian cells we use the G bending technique for the staining, and we separate the chromosomes on the metaphase stage of their division. So as I just mentioned in the previous slide, that the chromosomes they are color bodies, so that means they should be stained certain with the help of certain dyes. So they have chromatin. I have previously shown you in previous slide chromatin, and that is composed of protein and DNA, and that is its position could be in the center or somewhere away from the center. So this is very important that the cell, when they are not in the dividing phase, we cannot see the chromosomes probably because the chromosomes they are decondensed. Only during the mitosis or the division of the cells, the chromosomes could be possibly condensed and we can see under the microscope. So different species we know that they do have the different number of chromosomes. For example, in human we have 46 pairs or 46 number or 23 pairs. Similarly, cow we have 30, and that is the pairs. And similarly, different species the number could be different. Human and some other species, they do have the same number of the chromosomes, that's the 46. Well, karyotyping is basically the analysis of the chromosomes, a pictorial display of the metaphase chromosome from a mitotic or dividing cell. That is the basically karyotyping. While the cytogenetics is the study of the chromosome and inheritance. The cytogenetic, most of the studies are based on the human and Drosophila and some few of them some other organisms. Drosophila is fly and it's easy to use as experimental animals for study of the chromosomes. And human, the everything revolves around the human. All the studies, their focus is the well-being of the human. So human is also studied. Well, preparing for a karyotype, so we harvest the cells. That is the first cultured in the petri dish in the laboratory. Then the cells are treated with the colchicine, which arrests the cell in metaphase, and then treated and stained to observe the chromosome. So these chromosomes can be analyzed, visualized with the help of the computers connected to the microscope and with the specialized software. The chromosome could be identified by its size, position of the centromere, and bending and staining of regions. While this slide presents the preparation of the chromosomes, so in first stage, if you start with the left side, where a petri dish or the culture flask is shown in which the cell is being cultured, we add the tissue sample or we add the cells in a growth medium. It could be any one of the growth medium depending upon the type of cells. For example, M199 or some other media, and we add the chemicals, colchicine to stimulate the mitosis, incubate for 2 to 3 days so that the cell division is going on, and then add the chemicals that will stop the mitosis in metaphase. Transfer them, these are cells, to the centrifuge, uh, and you can get the cells after centrifuge, and then you can add the fixative material to fix the cells, and those after fixation could be transferred to the uh, plates or the slides, uh, and put on the cover slip, and you can add certain strains to enhance the chromosomes visibility, and under the microscope we can see the chromosomes, and with the help of the computer connected to the microscope we can see the pictures of the chromosomes. Okay, the previous slide, uh, this one. So this is what we see exactly under the microscopes, and from these shapes, different shapes, we have to identify the chromosomes by number, and this is the after arranged form from chromosome 1 to 22 and chromosome X and Y. Here is the normal karyotype of the cattle. I told you there are 30 pairs including the sex chromosomes. So this is all for this lecture. I'm keeping it smaller so that it could be easily uploaded on the uh, YouTube channel. And the next few slides in the subsequent lecture. We would uh, study certain international standards to name the chromosomes. Until that, thank you very much. Stay home, stay safe, and please go through all these slides, all these lectures through the YouTube channel so that you guys can get maximum benefit out of it. Have a good time. Thank you.